Welcome everyone. It's our sixth already, I guess, Journal Club meeting. And today the presenter is Dr. Ali Akbarinejad. Sorry if I spell it wrong. He's a postdoc at the Department of Chemistry and one of our most active collaborators from this department. Ali will present a paper of his choice today, and it's titled Engineering Extracellular Vesicles with the Tools of Enzymes Prodrug Therapy. Feel free to post your questions in the chat, and Ali, if you don't mind, we will stop you if the questions comes up and ask them. Yep. Cool. Off you go then.
Okay, thank you, Anastasia, for the introduction. Hello, everyone. My name is Ali, and as Anastasia mentioned, I'm a postdoc at the School of Chemical Sciences. And today I would like to talk about a paper which I talked about that with Cherry a few weeks ago, and we thought it's kind of could be a cool paper for this journal club to be presented. So let me share the screen with you. So can you please see the presentation file now? Okay, good. So, okay, let's get started with this paper. So, as you see in the screen, this is the title of the paper, Engineering Extracellular Vesicles with the Tools of Enzyme Product Therapy. This paper has been published in Advanced Material Journal in 2018. And I hope there are some kind of cool stuff in this paper that I can share with you guys. So, it seems that my computer is not working and it's not going to the other slide. Okay, let me stop sharing and share again. Hopefully it will work this time. Can you see the screen now? Okay. Can anyone hear me? Okay, good. Yeah, so it seems that it's working well now. So yeah, let's get started with the prodrug therapy concept. As many of you may know, prodrugs are compounds that are inactive in this format, so they are not toxic or they are not active in the body until they are converted to the real drug, the final drug. And usually this prodrug molecule has so this prodrug molecule has a kind of a linker, and this linker kind of makes this compound inactive. And after the prodrug being converted to the final drug with the kind of external force such as acid or enzyme or hypoxia conditions, it gets active. And for example, if we use this kind of a strategy for cancer therapy, this final drug can be toxic to the cancer cells and can kill the cells and do the job. So what is the goal of actually this paper? The focus of this paper is to make enzymatically active EVs and also liposomes, and then load these EVs into hydrogels, and then use the hydrogels to deliver drugs deliver the therapeutics into the kind of the cells and the point that they're going to use it. And as you see in this figure, they have some hydrogels, and this kind of black areas in these hydrogels show the EVs, and the EVs actually have been loaded with enzyme and with an enzyme. So the role of this enzyme is that it can convert a prodrug to the final drug. The prodrug here is a curcumin glucuronide, and the enzyme can make curcumin from this prodrug, and this curcumin has kind of anti-inflammatory responses. And what is the reason actually for loading enzyme into the EV? The reason is that these EVs or liposomes can protect the enzyme from degradation. So you all know that enzymes are vulnerable to the body conditions, and the aim of the paper is that protect enzyme from degradation. And here you can see the chemical structures of the prodrug and also the final drug. As you see in the prodrug, there is a kind of arm here, glucuronide arm here, and this arm makes the prodrug inactive. But after the enzyme cleaves this bond here, they can get the final drug, which is active. So before talking about the experimental conditions of this paper, I would like to show you this nice figure which shows different strategies for loading drugs into EVs. And we all know that EVs consist of a lipid bilayer which encloses an aqueous core. So, and this provides some spaces to load stuff into the EVs. And in the part A, you can see that if you have a hydrophobic drug, you can think about loading this drug into the lipid bilayer. And if the drug is hydrophilic, it can diffuse inside the EV and be loaded there. And if there are DNA and RNA stuff, and as you can see in C, they can also be loaded inside the EV. And also the EVs have a couple of receptors on their surface that can be used to label EVs with different stuff, with imaging reagents, with targeting agents. And in this paper, they want to actually load enzyme inside the EVs. So to do this, there are a couple of methods that people use to load cargo inside the EV. And they are usually classified into two categories. First category is passive methods, and in this method they just simply mix the drug with the EV and let the drug to go inside the EV, or they can add the stuff that they're going to load into the EV. They can add it to the donor cells, and in this case that compounds can be loaded into the EVs. And there are active methods as well, which are a couple of these methods: sonication, extrusion, freeze and thaw, electroporation, and also using compounds that can make the EV membrane kind of permeabilize the EV membrane. And for this aim, usually this compound saponin has been used, and this compound also has been used in this paper to load the enzyme into the EVs. So let's start with the results of this paper. So for the EV isolation, they isolated EVs from human mesenchymal stem cells using the normal ultracentrifugation techniques. And for loading enzyme, they just mixed EVs with the beta-glucuronide, which is the enzyme, and also saponin, which permeabilizes the EV membrane. A simple incubation for 10 minutes, and they say that it's enough for the saponin to do its job and load the enzyme into the EVs. And in the next step, they try to isolate the EVs and purify the loaded EVs, because you know that at this stage they have a complex mixture of EVs, enzymes, saponin, free EVs, loaded EVs. So they needed a technique to isolate the loaded EVs, and they used size exclusion chromatography to do this. And after they get the purified EVs, they can use it for their applications. And also, they tried to load enzyme into liposomes, and which are kind of synthetic vesicles. EVs are natural, and these liposomes are synthetic, and these synthetic EV vesicles have been used a lot in the literature. So that's why they wanted to compare the results with these synthetic vesicles as well. So, and it's interesting that they use a different actually technique to load enzyme into the EVs. I don't know what's the reason for using a different techniques, but as you see here, they use extrusion actually to do this, and to do this actually they mix the liposomes with the enzyme, and then pass this suspension through a membrane with very tiny pores, with 200 nanometer pores. And in this case, I think the membrane can be in touch with maybe the membrane, the channels on the membrane, because these channels are very, very tiny. And then it can kind of help the enzyme to be loaded into the particles. And then in the next step, they use the same column chromatography to separate and purify the loaded liposomes. So in this slide, you can see the size distribution profile of the liposomes and EVs. Figure A shows the size distribution profile of EVs and B liposomes. And as you can see, after loading the enzyme into the vesicles, there is a slight shift to the lower particle sizes for both of them. I don't know if this is a big shift in terms of the kind of resolution of the machine. So I don't know if they repeat the experiment, they're gonna see the same shift or not. But what we see here is that there is a shift to the smaller size particles. That, again, I actually don't understand why this should happen. But anyway, this is their results. And so in the next step, they tried to show that the enzyme has been loaded into the vesicles. They used a fluorescent system to show this. And we all know fluorescein. Fluorescein is a kind of a strong fluorescent compound. And there is a derivative of fluorescein, which you can see in the left, which has again that glucuronide arm on the molecule. And this glucuronide arm again kind of makes differences to the whole structure, and this structure actually is not fluorescent. And this enzyme that is used in this paper can again convert this glucuronide derivative to the final fluorescein compound, and then make the fluorescence. So they used actually this fluorescent system to show that enzyme has been loaded into EVs. So what they did is that they loaded the vesicles into a hydrogel, and put the hydrogel into the non-fluorescent version of the fluorescein, and give it some time to make the final fluorescent compound. And as you can see here, there are some fluorescent signal from both EV and also liposomes, which shows that enzyme has been loaded into these vesicles. And so very, very big actually error bars. So they say that they have kind of comparable loading efficiency for EVs and liposomes, but again, I don't know if this is a maybe good conclusion, because the error bars are quite big, and I think it's hard to decide about the loading efficiency based on this kind of results. So in the next step, they try to load the vesicles into into the hydrogels and use them for their kind of therapy, cancer therapy, not cancer therapy, therapeutic applications. So they used this hydrogel, PVA, hydrogel of PVA and PEG to load the EV and liposomes. And as you see in this figure on the top right, they made a couple of hydrogels, a hydrogel with the free enzyme, a hydrogel with EVs, and this EVs for sure how enzyme inside them, and hydrogel with liposomes and the control hydrogel without loading anything into that. And they used confocal imaging to show that EVs have been loaded into the hydrogels, and as you can see, there are some spots with a strong green fluorescent, which shows that EVs are there, but for sure, these spots cannot be separate EVs because the size is quite big. And in the paper also, they mentioned that they don't have separate EVs. They have areas which are enriched in EV. And again, they didn't provide any reason for observing this kind of structure. And they did this in 2 different concentration of EVs. And as you see, for the higher concentration, there is more of this fluorescent spot. And they also used a kind of new technique for imaging the EVs, as you see on the right, which I'm not going to go through the details, just to say that the name of the technique is density-dependent color scanning electron microscopy imaging. And it seems that in this technique, they used some urinal label EVs, and this urinal label EVs kind of can be visualized very well with this technique inside the hydrogels. And they say that this is actually the first application of this method to visualize the EVs into the complex mixture like a hydrogel. So in the next step, they studied the enzyme release of the hydrogels because actually the aim of this project, actually ideally they want to keep these enzymes into the hydrogels. So they don't want the enzyme to leach out from the hydrogels. So they did this experiment again with different hydrogels. With as you see in this figure on the top right, they have non-loaded hydrogel. They have EV hydrogel, liposome, and also hydrogel loaded with the free enzyme. And the result that they got from doing these experiments is that when they have hydrogel, which is loaded with a free enzyme, they get a higher fluorescence intensity in the solution. So I should add that they use the same fluorescent system of fluorescein to show that to do these experiments. So they have these hydrogels, and then they put the hydrogel into the solution of the fluorescein derivative, and then the enzyme can do its job and make the final fluorescein, which is kind of fluorescent. And as you see, we have a kind of higher fluorescence intensity for the hydrogel, which is loaded with the free enzyme. And this shows that the EV and liposome can help the enzyme to kind of stay in the hydrogel and don't leach out from the hydrogel. But there is something here that, again, I cannot understand. And this thing is that if you look at the hydrogel, which is not loaded with anything, still we have a kind of fluorescence intensity which is comparable to the EV hydrogel and liposome, and also it's just a bit smaller than the other hydrogel which is loaded with the free enzyme. So what I expected here, that we observe a very, very low fluorescence signal for the hydrogel which is not loaded, but there is some signal from that sample as well. But anyway, the hydrogel loaded with the free enzyme shows higher fluorescence intensity, which I think can show that EV and liposome can help the enzyme to stay kind of more tightly in the hydrogel structure. So the next study is studying the enzymatic activity of EV and liposome-loaded hydrogels. So, or actually over time to see that how kind of active is the enzyme there. So ideally, they want to see kind of prolonged enzymatic activity for the hydrogels. So if you look at the figure A, again, I should say that again they use the fluorescence system to do this study. And if you look at the figure A, you can see two different columns: the red columns, the red solid columns, and also the pink patterned columns. The red columns shows the fluorescence intensity for the incubation of the EV hydrogel into the fluorescent solution, and then measuring the fluorescence signal. And as you see, when they do this over time, overall there is an increase in the signal, in the fluorescence signal, which is something that we expected. And actually, to see how stable and how stable is enzyme there, they take out at the end of day 7. They do this for 7 days. At the end of day 7, they take out the hydrogel and then put it into a fresh solution of fluorescent compound. And then again, they measure the fluorescence intensity over time. And as you see, if we consider the day 7, for example, there is a decrease in the signal for the second round. So actually, there is 29% decrease in the signal. If you look at the figure B, these results are for the liposome hydrogel. And in the paper, they say that at day 7, they can see 96% of the activity of the enzyme at day 7. But what I see here is that at day 7, for the second round, there is even a kind of more intense signal than the first round. That again, we don't understand why this can happen. And then for the hydrogel which is loaded with the free enzyme, you can see that for day 7 and also for day 5, there is about 77% decrease in the activity. So as I said, it's difficult to find the kind of trend for these results, but overall we can see that for the hydrogel which is loaded with the free enzyme, there is kind of more decrease in the signal after incubating the hydrogel with the solution for a long time. We can maybe show that the EV and liposome can help the enzyme to be protected from degradation. And finally, they use the hydrogels for therapeutic applications. And to do this, they used these cells, mouse macrophage cells, and they challenged these cells with a bacterial lipopolysaccharide that they say that I'm a chemist, I don't understand the biology very well. Please correct me if I'm saying something wrong, but it seems to me that they incubate these cells with this lipopolysaccharide to make a kind of inflammatory conditions with these cells. And after that, they have these cells with with inflammatory conditions, and then they add the curcumin beta-glucuronide to this substrate, and after this, if they add the hydrogels, the hydrogel is gonna make react with the beta-glucuronide compound and make the curcumin released from this compound, and then curcumin has anti-inflammatory properties, and it can, from what I understand, is that it kill these cells and reduce the cell viability. So if we look at figure A, you can see the cell viability results for different hydrogels, so you can see that there is kind of very low cell viability for the EV hydrogel in red, for the blue hydrogel, which is the liposome hydrogel, for in the blue column, which is the liposome hydrogel, and also the green column, which is the hydrogel, which is loaded with the free enzyme. And if we compare this cell viability results with the cells alone, and also with the hydrogel, which is not loaded, it's kind of clear that this kind of hydrogels have decreased the cell viability. And it shows that the enzyme is active. But the question here is that if the hydrogel itself can kill cells and decrease the cell viability, what is the point of using EV-loaded and liposome-loaded kind of liposome-loaded hydrogels? So actually the point here is that when we use EV or liposomes, it can protect the enzyme from degradation. So they expect that if they do this experiment for a long time, then the activity of the enzyme will decrease for the hydrogel which is loaded with the free enzyme. But then when they have EV and liposome hydrogels, the activity of enzyme should be kind of still, the enzyme should be still active for a long time. So if we look at figure B, you see that the EV and also liposome hydrogel still kind of have low cell viability, resulted to low cell viability results. And for the beta-glucuronide hydrogel, which is hydrogel with the free enzyme, the cell viability is increased. And they say that actually after they did this for 7 days, this figure showed the results for 2 days. And they say that after they did this experiment for 7 days, this hydrogel with the free enzyme completely lost its activity. And after 2 days, you can see that there is some activity, but it's definitely less than the kind of initial results. And so yeah, I think this figure shows that EV and liposomes are effective in protecting the enzyme from degradation. Yeah, that's it. This is almost all the results. Just a quick conclusion. So in this paper, they reported the first application of EV hydrogels for delivery of anti-inflammatory drugs, for the kind of controlled delivery of anti-inflammatory drugs. And they also encapsulated enzymes into the EVs and liposomes, and they showed that the EVs and liposomes can somehow protect the enzyme from degradation and make the enzyme to have prolonged biological activity. And the other result that they have is that the kind of anti-inflammatory potential of EVs is comparable, or in some cases even better than the synthetic liposomes. Yeah, it's all that I wanted to say. Thank you very much for listening, and I would be happy to take the questions.
Thank you very much, Ali. Really interesting presentation. Yeah, I enjoyed the paper of your choice. We have a couple of questions in the chat. So the first one is from Vanessa. She asked if they used anything else rather than PKH 67 as a control.
I don't think so. I don't think so. For the confocal experiments, they just use PKH 67 to take the images. And of course, they used another technique to image the EVs, but for the confocal, they just use PKH. Yeah.
There was a comment from Sheree. Sheree, sorry, I don't understand what that means.
Okay.
I didn't really have any comments. I was just chatting as I go. Yeah, the error bars. There was very big error bars throughout.
Very big. Yeah.
Yeah, I was like, really? Do you think that's down to the variability in the loading of the hydrogels because it had that clumpy vesicle look, or whatever it was, my cells probably. It had that clumpy look, and I wonder whether the loading of those things is really difficult, and maybe that's what the problem was with the error bars.
In the loading of EVs into the hydrogels, or loading enzyme into the EVs?
Well, yeah. It's whether the replicates are the loading of the vesicle, the drug into the vesicles, and then the vesicles into the hydrogel, or whether it's the same batch of loaded vesicles into. It's whether who knows how they designed it. But
yeah, I think from the beginning, actually, which is loading the enzyme into the EVs, there are some things that I don't understand very well, actually. Why they use different techniques to load enzyme into the EVs and liposomes. So because I think they have the same size range. So in terms of size range, there is no reason for using different techniques, right? So what I think is that maybe yeah, the loading efficiency for maybe some of the techniques were not that successful, and then they decided to go and try the other methods as well. But yeah, and for those results, if you look at the kind of error bars, they are very, very big, actually. So when they compare the loading efficiency of the enzyme into the EVs and liposomes, the error bars are actually huge. So yeah, and it's interesting to me that they had a conclusion that the loading efficiency into EVs and liposomes are comparable. But I think it's hard to make any decision based on kind of results with those very big error bars, because I'm sure that if they do a statistical analysis, they are not significantly different, for sure.
I was worried about. I was amazed by the density dependent color scanning EM as well, and so I had a Google, and Colin said that he can just do EM and add colors in Paint, and it seems that is actually what you do. Pretty much. So I was like, oh, that's really exciting. We should do that. Okay, maybe not.
Yeah, actually, I didn't want to talk a lot about that, actually, because I was not convinced with those images, actually. So maybe I can try to share again with you that.
I think one of the problems with all of the imaging is the resolution of trying to see tiny dots.
Yeah, for sure, the resolution is not enough for the confocal. It's not enough to see the single vesicles, and for sure those kind of separated spots are not vesicles. But so what I expected is that we see a kind of a background of green color in the kind of hydrogel, because yeah, I don't understand why they should see kind of areas which are enriched with vesicles. So I don't know, maybe EVs tend to aggregate, something like that, when they are into the
They do. If you're isolating through ultra, then they will clump together, and the dye I don't think helps either. So, and then you're mixing it with a hydrogel to make it. Yeah, I suspect it's just a really hard thing to mix up.
Yeah. So can you see the screen now?
Yeah.
Yeah. So yeah, for this density dependent color scanning imaging, yeah, I was not totally convinced, because if we compare the left image with the right image, the left one is the EV hydrogel, and the right one doesn't have any EV, and even for it seems that they look at those red spots as the EVs, right? And then for the right one, we also have those red spots. So yeah, I don't.
The main difference I can see is that the EV loaded one is furrier for green, and the other one's smoother.
Yeah.
Very
Yeah,
good
but
scientific interpretation.
yeah, but for those points that they show with, with these arrows, yeah, I think it's hard to say that they are EVs because we have them in the right image as well, maybe somehow.
Do you think the PVA pig hydrogel, do you think changing that will make a huge difference on release as well?
You mean the release of enzyme?
Yeah.
I think for sure it can affect, yeah, because for sure maybe not the enzyme alone when you have the enzyme loaded into the EV, because in this case enzyme is not in touch with the hydrogen, right? So in this case that's the EV which is in touch with the hydrogel. So I think it will depend on the interactions of the hydrogel and EV, and not enzyme and hydrogel. So and I think it can definitely affect. If you look at the you know, those functional groups that are present in the hydrogel and also the surface of the EV, for sure there should be different interactions between them. And if we have a hydrogel which has kind of stronger interactions with the EV, it should help to kind of keep the EVs there.
Which, yeah. So you'd think that the loading between EVs and liposomes will be quite different considering the coating. It's an interesting challenge. There's probably 100 ways, 100 different types of hydrogel that you could try.
Yeah.
As well as 100 different ways of encapsulating your enzyme. It's interesting.
Yes, yeah. So actually, this is the way that they did this experiment, that they loaded EVs into the hydrogels, and I think the reason is that maybe in this way they can just take that hydrogel and put it in the place that they're gonna do the experiment. So, but the other way is that you just use the EVs, right? And then try to surface functionalize them and try to deliver them. And this also could be maybe a good way to do these experiments. You know, make EVs, load stuff into them, and then try to deliver them. Yeah.
I think the value of these is the single dose slow release concept.
Yes. Yeah.
Quite fun. I think we should start looking at this.
But not releasing, actually, not delivering the EVs, because in this case, EVs, they say that EV is trapped there, and it's just the final EV is trapped, and also the enzyme is trapped, and it's just that compounds that can diffuse into the gel and also go out. Yeah.
So it seems a shame. It seems actually a shame because your vesicle has got these superpowers already, being anti-inflammatory, especially the mesenchymal stem cell ones, and you're not even using that. You're just a package.
Yeah.
Do we have any other questions to Ali? Nothing in the chat either. In this case, thank you very much, Ali. That's a really interesting paper.
Yeah, thank you all for listening to the presentation, and I hope it was useful.
It was, yeah. Really good one. Before we leave, we have a couple of more announcements for today. So first of all, we will still continue to have our journals clubs under the alert level 2. So that means if you want to present, and we really encourage you to do so, please contact me, and we arrange a next spot for you. Next week we will have Dr. Sandy Lau presenting her paper of choice, and based on today's poll, most of you would like to see a mixed type of this meeting. So feel free to include your results to this presentation. You can present both a paper and your results. So it's really, really up to you now to decide how you want this presentation to look like. And another important announcement, so you all may know that this year ICEF meeting would be virtual with a lot of live talks and pre-recorded presentations. And we are very happy at HEVI to announce that we will be supporting applications, and we are calling for expression of interest to attend this virtual meeting for the people who are based at the University of Auckland. So please look for the emails. They will come out shortly, hopefully this week or early next week. And you still can apply for the poster presentation. It will have really cool interactive format when you will have 3 minutes to present your poster in a format of Zoom. Correct me, Sherry, if I'm wrong here.
It's a little cadmium platform. So you make a presentation in landscape mode, and then you record over it online, and you can, as far as I know, you can zoom in and show, oh, look at this graph here, and then you can zoom out again, and you can move to the next one. So it should be quite a fun, interactive poster session, all available for 60 days afterwards as well. So it's on demand for 60 days. So huge value for money. Not that I'm plugging it.
So yeah, look carefully for these emails in a couple of coming days and apply. Yep, and we hope to see your applications there soon. Thank you very much for joining us today, and we will see you next week, as always, at 2 p.m. Thank you all for coming.
Bye-bye.
See ya.
Thank you. Oh, yeah.